In this activity, we're going to look at apical dominance, which is the pattern where the terminal bud at the tip of a plant suppresses the growth of lateral or axillary buds. This is a major way plants control their branching pattern and overall shape, and it turns out that auxin plays an essential role in this process. Many plants show apical dominance, meaning the terminal bud at the top of the stem keeps the lateral buds from growing. This suppression is largely due to auxin produced in the apical meristem. When the terminal bud is removed, that source of auxin is gone, and the lateral buds are released from inhibition and begin to grow. If auxin is then reapplied to the cut stem, apical dominance is restored, and lateral bud growth is suppressed again. This shows that hormones play a direct role in shaping how a plant branches. Apical dominance has big consequences for plant form and survival. It affects how tall a plant grows, how much it branches, and how it competes for light. It's also something growers use all the time. When you prune a plant, you're often deliberately breaking apical dominance to encourage branching. So this isn't just a lab concept. It's a real practical part of plant growth and management. Before looking at the experiment, think about this question. Why would it be useful for a plant to focus growth on one main shoot instead of branching right away? Consider things like height, light capture, and competition with neighboring plants. Write your prediction in your lab notebook. You'll come back to this idea when you interpret the results. In this experiment, similar plants of the same species and growth stage were used. One group was left intact with the apical bud still in place. In a second group, the apical bud was removed and no hormone was applied. In a third group, the apical bud was removed and auxin was applied to the cut surface of the stem. All of the plants were grown under the same environmental conditions, so the only differences were the presence of the tip and presence of auxin. After a set growth period, the plants were examined for lateral bud and branch development. The presence and extent of lateral growth were recorded and then compared among the three groups. This made it possible to directly compare how removing the tip and adding auxin back affected branching in otherwise similar plants. Now you'll use what you observed to answer these three questions in your lab notebook. First, summarize what happened in each treatment group. Then explain why the lateral bud started growing after the tip was removed. Think in terms of hormone signals. Finally, step back and think about the bigger picture. How does apical dominance influence the overall shape of a plant and its growth strategy in the environment? The key takeaway from this activity is that auxin from the terminal bud suppresses lateral bud growth. When the tip is removed, lateral buds are released and begin to grow. When auxin is added back, that suppression returns. This is a clear example of how plant hormones regulate branching patterns and overall plant form, not just how fast a plant grows.